Thanks a lot, Nadia, for this nice introduction, and welcome all of you to the last session of today. And as Nadia mentioned, my name is Dominik Witzigmann, and I'm from the University of Basel in Switzerland. And today I will talk about targeted DNA therapeutics for hepatic diseases. And I would like to start with a clinical case to give you some impression about our vision, why we want to develop plasmid DNA therapeutics. So this is the 9-year-old girl, Brianna Minek, and she is diagnosed with one of the rarest diseases in the world. It's called Crigler-Najjar syndrome type 1. So the problem is with this genetic disease, she cannot, or the bilirubin accumulates in eyes. You can see her yellow eyes in skin and in the fatal phase, also in the nerves, in the brain, and then can lead to death. And all is due to a genetic disease. And the only available treatment option at the moment is a phototherapy for 10 to 12 hours, which helps to metabolize the bilirubin and therefore, make the life a bit better. And this is just one example of a hepatic disease which is really severe and where there's a lack of efficient therapeutic options. And recently, 5 months ago, I also faced these problems, or have seen the huge list of hepatic diseases. So 5 months ago, my son was born, and the usual procedure when a child is born is you take blood samples, the dry blood spots get analyzed for genetic screening, and then they look for genetic diseases. Luckily, there was no genetic failure in our son, but I was interested, what is the case in other countries? And then I checked, and in Switzerland, they test for 9 different diseases. In Germany, for 12 different diseases. And in the US, you see the list is quite long, 61 diseases. So I asked myself, what is the reason for this? And the reason is, the Swiss policy is, we detect what we can treat. And the US policy is, we detect whatever we can detect. So you see, there's a clear lack of efficient therapeutic options. And there is a nice article in Lancet which shows the UK liver disease crisis, but this holds true for the whole Western society. And they wrote, survival rates have improved for almost every disease of every organ in the last few decades, with one notable exception, liver disease. And the problem is, we have a lot of technologies and also gene editing systems, but we are facing the problem that we don't have the right carrier systems, and they are not efficient enough. So we need carrier systems to specifically target the right cell type. And the idea or the vision we have in our group is to use plasmid DNA delivery to the right cell type within the liver. And we had a nice talk from Carla. She was mentioning that liver targeting is not just liver targeting. There are different cells. So we have the blood vessel in the middle, surrounded by the sinusoidal endothelial cells in yellow, and the main enemies of nanoparticles, the Kupffer cells, then the stellate cells, and our target, the hepatocyte. So what we aim for is a nanocarrier system with the plasmid DNA, passing these liver sieve plates you see down here, they have a size of around 100, 150 nanometer, and bypass the Kupffer cells, entering the hepatocyte, or specifically bind to the hepatocyte and bring the plasmid and DNA into the cell, and therefore have a long gene expression. So not just a short expression for weeks, so expression for months. And there are many hurdles, and we stepped over several hurdles during the last years. And I want to focus on the first few hurdles. So the nanoparticle formulation, then we have the circulation in the body, then the escape from the blood vessel through the fenestrations, then the binding to the hepatocyte and uptake. And then there are other hurdles, like Steve mentioned, the access to the nucleus and then expression of the protein. And in my talk, I want to focus on the 4 main important factors we are thinking are the important factors to develop something like this. Is first, the nanoparticulate drug delivery system, then the ligand to target the right cell type, the target receptor, and the in vitro, in vivo models. We will see this in the upcoming slides. So I will always start in the upper left corner. You see which topic I'm facing. So the nanoparticulate drug delivery systems. In clinical trials, there are many different carrier systems, and our group is focusing on lipid and polymer-based nanoparticles. And for this project, we use multi-component lipid formulations, like in many research groups in the siRNA or mRNA field. So we have a structural lipid, and cholesterol, a PEG lipid. Then we have, together with the University of Fribourg, we design aminolipids for the conjugation or encapsulation of plasmid DNA. And we have a ligand PEG lipid. We conjugate the ligands via malamide chemistry to the surface or the distal end of the pegylation. And the nanoparticle formulation method is one crucial step in this whole procedure. And we are using the microfluidics approach because it's rapid, robust, and there's possible scale-up. And here, one example of a POPC-based formulation. So what we do is usually a design of experiment. We variable the total flow rate and the flow rate ratio you see in the left side, and the size. And then with our software, we can analyze the procedure, and then we can directly see which sizes do we get when we use a certain total flow rate or flow rate ratio. Or in the middle, you see the PDI. Because the reason is when we compare different nanocarrier systems, so different aminolipids, we don't want to compare apples and cherries. We want to compare the same size. And this is important for in vitro, but also for in vivo studies. And on the right side, you see an image of these particles of the multilamellar structure. So we think the nanosampler benchtop instrument we have in our group is a really perfect system to screen a lot of different formulations really fast and therefore compare the size and the different settings we need. Just a small intro into the in vitro study we are performing. So when we do the in vitro or the production of the nanoparticles, we then use the different nanocarriers and look to the gene expression using different ratios or different components in the media, and then look for the gene expression. And then we also use a design of experiment to really see what kind of influence does the N to P ratio have, or the amount of DNA we use. And we see a strong correlation between the formulation aspect, so how we prepare the nanoparticles, or what components do we include, and also on toxicity. Then the second question is, when we have our nanoparticle, what target receptor do we use? And for hepatocyte-specific targeting, there are many receptors, mainly in the siRNA or mRNA field. The people focus on the LDL receptor. In our group, we are taking the asialoglycoprotein receptor as a hepatocyte-specific target. It's a C-type lectin, which binds multivalent sugars, like you see in the left panel A. And this receptor is abundantly expressed on hepatocytes and predominantly expressed, which is a really good target to target the right cell type. And after binding, there's a clathrin-mediated uptake into the cell and therefore brings the payload into the cell. When we started this idea or this project, we asked ourselves, what is the situation in clinic? So together with the pathology in Basel, we evaluated an RNA and tissue microarray screening and looked to different patient samples and the expression of the asialoglycoprotein receptor. And what we have seen on the left-hand side, you see the non-tumoral healthy liver, there's a high expression. But what we have seen is that the expression shows a high variability between the different patients. So when you look here in the audience to your neighbor, I'm sure your neighbor has or your person next to you has a different expression level of the asialoglycoprotein receptor. An interesting thing was that in cirrhotic specimen, we have seen that there's an increase in the expression level. And a bad thing was in the tumor liver, there's a decrease in expression level, even with increasing Edmondson grade, so with increasing stage of HCC, which means this target receptor cannot be used for HCC therapy. But in the non-tumoral healthy liver, and we also have some samples from genetic diseases like alpha-1 antitrypsin deficiency, we see a high expression, so it's a good target. Then I will come to the ligands. First, the in vitro, sorry. So the systems, when we have the systems, we want to test them in vitro. So what we did was a screening of the different cell lines in our lab, liver cell lines, and looked for the expression level, and we also have seen that there is a huge variability in the expression level of the asialoglycoprotein receptor. The highest expression was found in HepG2 cells, and we have seen a nice correlation between mRNA and protein level, and this holds also true for the tissue microarray stainings in human beings. The nice thing about the asialoglycoprotein receptor is that in vivo models are available. This means mice and rats, they also have the same receptor and they recognize these multivalent sugars. So now I come to the ligands. there are many ligands available how you can target this receptor. You can use monovalent sugars, complex carbohydrates, glycoproteins, or antibodies. All have advantages and disadvantages, and I will mainly focus on the middle two. But what we have to test, or what we always tested, was what is the affinity, and especially for the sugar ligands, it's important because there is this cluster glycoside effect, so the simultaneous binding of more than one sugar, so 2 or 3, increases the affinity or avidity very much, so it gets more specific, the binding, and triggers the uptake. Then an important thing we have realized is the ligand density. You really have to balance out the targeting and the loss of the pegylation, so the stealth effect. And also the linker length plays a crucial role, that there is no steric hindrance in cell binding. So I will focus on the complex carbohydrates and glycoproteins, just 2 studies I want to mention. The first one is the complex carbohydrates. So for this binding to the asialoglycoprotein receptor, only the sugar moieties are important. So what we did is we used an enzymatic reaction and an extraction separation method to isolate these complex carbohydrates. In contrast, many groups use a synthetic way to synthesize these complex carbohydrates. The advantage of the natural product is you already have this spatial arrangement, which is really important for a specific binding. As a model compound, we used a fluorophore, and we could show that there is a high uptake, which is really receptor dependent. One example from an in vivo study using our targeted liposomes was the pegylated liposome you see on the right-hand side, and we conjugated asialofetuine, which is a ligand for the asialoglycoprotein receptor, to the surface of the liposome. And what we have seen that in the liver there's a huge uptake, whereas in the spleen there's only a slight uptake. When we compare this to the non-PEG liposomes, there's a huge uptake in spleen, so an organ of the reticular endothelial system, and only a punctuated pattern in the liver. And what we could show then was that for the non-PEG liposomes, the uptake in the liver are the Kupffer cells. So we did a co-staining with carbon plaque, and we have seen that these are not the hepatocytes. So it's the liver targeting, but the wrong cell type. And for the targeted liposomes on the row below, you see that the AFPEG liposomes showed a high uptake, as I mentioned, and we could competitively inhibit the uptake, so it's receptor specific. So I have shown you the 4 important factors we are focusing in our research lab, and I want to come to a summary and to tell you why combining targeting strategies and DNA's advantages. And what we think is that we need improved nanoparticulate drug delivery systems. And we use authority-approved formulation and modify them with our lipids. Then we need a method which is reproducible, scalable, adaptable. And we think that fast screening is really crucial for the development. And this, I think, the nanosampler benchtop is perfectly fitting for this. Then the hepatocyte-specific targeting strategies. You have seen that in healthy liver, there's a high expression. But what is important, that there is a variability between different patients. So we think there is a need of diagnostic tools if we use nanoparticles itself or also target systems. Like it's usually done in clinic when we talk about antibodies, for example, for breast cancer, HER2, EGFR receptor overexpression. Then the assessment of the targeted DNA therapeutics. We faced a lot of problems, and we really have to understand more how the nanoparticle gets uptaken and what is the intracellular trafficking. So sometimes we see an uptake, but we don't see any gene expression. So we really have to get clear about this. And I've shown you that we have, with our target system, in vivo proof of concept, but we're still facing the problems that the plasmid DNA comes to the cells. We see expression, but we would like to increase this very much. And in our work, we are collaborating with pediatricians, clinicians, and chemists to really drive this technology forward. So our vision is to have an alternative strategy to the light of life. So develop a plasmid DNA delivery system to have a long expression of a certain protein. And the financial support I want to mention is from Novartis, University Basel, and FAG Basel. And all this work would not have been possible with the collaboration and help, support of a lot of people. So my boss, Professor Jörg Huwieler, and a lot of talented PhD students and postdocs in our lab. and then our collaborative partners for the German cancer research centers, then the hospitals in Basel, Zurich, Heidelberg, the Zumbü lab in Fribourg for chemical synthesis, and the Uni Freiburg for help with the physical chemical characterization. And thank you for listening. I'm open for discussion, and in Swiss German we say merci vielmals, so thank you. And before I end up, I invite you to Basel. On May the 7th to the 10th, there's the CLINAM, the Clinical Nanomedicine Conference. It's a really perfect and interesting conference. I highly invite you to come to Basel to enjoy the conference, enjoy some Swiss chocolate and cheese, and Basel City. Thanks a lot.
So, danke vielmals, Dominik. Yeah. And now your questions. Gesine is jumping. Yeah.
Very nice presentation. I'm wondering if you have a strategy for the hepatocellular carcinoma cells. They don't express your receptor. Have you tested the non-targeted?
Yeah, so currently it's nice that you asked. This is perfectly fitting. So we have a project together with one of my colleagues. I have to say hello because my colleagues are sitting in the lecture hall with popcorn and watching me. So, and we have a project on HCC where we use at the moment a non-targeted delivery system for the expression of an oncotoxic protein. So it's undergoing. We are going in vivo in mice. So let's see. Maybe the next meeting I can tell you more.
Yeah, very nice, thank you. So regarding the different types of targeting moieties, maybe you can comment on what you think the pros and cons are of certain ones and which ones you think will be successful with Nanomedicines.
So the sugar moieties are I think the sugar moieties have much more advantage compared to the antibodies for this receptor because the sugars are not immunogenic. Then when we compare complex carbohydrates and glycoproteins, I would prefer complex carbohydrates because in the glycoprotein you still have the protein part which might be immunogenic. And if we compare monovalent sugars and complex carbohydrates, this depends really on the formulation method or the nanoparticulate drug delivery system because for the specific binding, sugar-protein interactions like we have here, this lectin receptor, is really weak. So we really need this specific conformation for the binding. So if we just have a polymer and then at some positions conjugate a galactose to it, it will not be very specific. We really have to think about this special conformation. Yeah.
I might have missed it. One of your targets was to express the DNA for long periods of time. Did you show any data on that?
No. So together with a collaborative partner, we checked the expression level of DNA, or they have checked the expression level, and when the DNA is entering the hepatocyte, it's for the lifetime of the hepatocyte. So they had to stop after 180 days, and there was still an expression. And the average lifespan of a human hepatocyte is around 150 days, which would be perfect, because if we could manage to enter the cell and bring it to an expression, we have a really long expression level of the protein.
Is this a normal plasmid that you're using, or you're using a special?
So we have normal plasmid, but also modified plasmids. Okay. And also we look to the promoter that we, for example, we have plasmid with hepatocyte-specific promoters to have another targeting strategy. Good, thank you.